So welcome everyone to our Zoom presentation with Dr. David Scher. We're excited about this opportunity for you all to learn about the science of prevention and research on how cancer can be stopped before it starts. I'm Drew Lindgren. I'm the Director of Community Engagement at Find the Cause Breast Cancer Foundation. So did you know that of all the funding for cancer research, only 7% goes towards prevention? Find the Cause is dedicated to changing that imbalance by funding research efforts that accelerate the science that we hope will lead to a safer world for generations to come. Imagine how different the world would be if we could prevent cancer before it ever happens. That's the focus of the Find the Cause Research Consortium led by Dr. David Scher. Dr. Scher received a BA in biology from Brandeis and a PhD in microbiology and immunology from Cornell. He's a professor of environmental health and professor of pathology and laboratory medicine at Boston University. He's the director of the BU's Superfund Research Program and the co-director of the Cancer Intercept Program at the BU School of Medicine. I could go on with the list of his many qualifications, but in the interest of time, we'll end with what we feel is his most impressive. He is the director of the Find the Cause Cancer Research Consortium, as well as a member of our board of directors. His work and the work of the other consortium members is to find the environmental causes of cancer, to energize grassroots efforts to stop the production of environmental carcinogens, and to develop approaches to intercept cancer before it starts. It's now my pleasure to turn the program over to Dr. David Scher.
Thanks very much, Drew. That's very, very kind and generous of you. I'm proud of that last title. So today I'm going to be talking about what the Consortium of Scientists does and being funded as they're funded by Find the Cause. So we have a consortium of 4 scientists. We have 2 of us at Boston University, myself and Stefano Monti, right here. And at Tufts University, Charlotte Cooper-Wasser and Gail Sonnenschein. Now, the idea behind having a consortium like this is that science moves too fast for any one laboratory to really advance very, very quickly. The knowledge advances quickly, the technology changes daily, and it's unbelievable technology is being developed right now. So the best way to do these things is to do it as a team. The 4 members of this consortium have a common interest in cancer, particularly breast cancer and preventing cancers. And they all have overlapping expertise, but unique expertise. So together, we can add things to each other's projects. We contribute ideas that one or another may not have thought of. It works much more efficiently as a team, as you'd expect. You get more than the sum of the individual parts. So, our overall tenet is that we're trying to use team science to unravel the biologic processes set in motion before cancer appears and to intercept it before it starts. So I've broken it down into 3 particular areas, which we'll talk about today. One is to identify carcinogens, and that's no small task. I'll show you what we're doing to do that. We're going to try to predict when cancer is going to happen. So it sounds a little magical right now, and maybe it is, but maybe in 2 or 3 years, it won't be quite so magical. And I'll show you some data at the end that shows that we're making pretty good progress in that direction. But then once you can predict that someone's about to get cancer, you have to be able to intercept it. So that's part of the program as well. Now, the paradigm of cancer, the way it's viewed these days, goes something like this. Let's say there's something in the environment that starts a cancer, and we can talk for a long time about the contribution of environmental chemicals, and we will, or the contribution of bad luck, not so much. But the way the establishment now sees things is that they will see you at this stage when they can do some imaging and detect something that looks slightly abnormal. Now, at this stage, it's either a precancerous lesion that's really about to blow up, or it's a cancer itself. And by the time you find this, it may already be too late. It may have already been metastasis. So something like ductal carcinoma in situ is, by some definitions, an early cancer, by others, a precancer. But what will happen is that they will, much of the time, physicians will either watch this for some period of time and wait for other things to happen, like more mutations, and then they will start treatment. some cases in some cancers, this will be removed, and some others not so much. Now, what we're trying to do is open up this big black box in the middle, which is what happens between something that initiates the cancer and something that you might think of as maybe it's already too late, a precancerous stage. So we have two particular goals. The first is to, well, identify which of these environmental carcinogens are causing cancer, and then use that as the power to prevent exposure to these chemicals. And then failing that, we want to be able to predict the cancer before it actually happens, and then to do prevention at this time. So we think of reducing the exposure to these chemicals as primary prevention, and intercepting it at a later time point, secondary prevention. So it's all about prevention. So I'm going to give you a little bit of good news and bad news, and good news and bad news, and here's what that looks like. In the US alone, about 1.7 million new cases of cancer will be diagnosed in 2019. Well over 500,000 people will die. The economic cost of cancer in the US is about $95 billion per year, so it's a big problem. From 1991 to 2015, cancer death rates have decreased by 23%. That's great. Death rates have decreased. However, cancer cases are increasing. So in the next 15 years, there'll probably be a doubling in the number of cancer cases. Doing a little bit better on the treatment part, not so good on the prevention part. And it's clear, you look at the numbers, we can't economically or scientifically treat our way out of this. I mean, you just can't do it. It'll overwhelm the healthcare system. The good news is Find the Cause, which is one of the only organizations that's focused only on environmental causes of cancer. Our donations have increased by about 50% last year. So we're doing well, and people are beginning to get the point, and we're beginning to get the kind of support that's really needed to do prevention. Less than 7% of the NIH budget goes towards prevention. That's the bad news. So here's a graphic that represents all of the institutes of the National Institutes of Health. Here's the National Cancer Institute. That's the big gorilla in the room. Its budget is about $6.1 billion. Now, this is a guy named Norman Sharp, who is the director of the NCI. He came to visit us at BU about a year or so ago. I sat down with him and he said, yeah, we're doing so much now for cancer. We're making great progress. I said, well, what about prevention? And Dr. Sharpless said, yeah, we're doing prevention. We're spending X amount of dollars to improve our imaging so we can detect them early. I said, yeah, detect them early. That's great. What are you doing to prevent? I mean, what are you thinking in the future? Oh, in the future, we'll have more and more technologies to be able to identify this stuff early. The stuff being cancers. I said, and after asking the question 3 or 4 different ways, I finally gave up and decided he was not going to start thinking about prevention as actually preventing something bad from happening, which my definition is you have prevented some person from having to go from their physician to their house, to their family and say, I went to the doc today, I got bad news, I have cancer. We're trying to prevent that entirely. You also notice some of these other institutes. This is the National Institute of Infectious and Allergy and Infectious Disease. You'll be amazed that I was able to find a photo of Tony Fauci on the web. So here's Tony. All right, the good news is 60 to 70% of all cancers are preventable. There's been a lot of dialogue going on in the scientific field about how much, whether it's 60% or 80% or 70%, it's somewhere around there. So that's good news in a sense, because it means that we're able to identify things that are causing cancer and stop it. If it weren't preventable, we'd all be talking about treatment. Now, you'll know that I'm starting to segue over into the effects of environmental chemicals. So how big is the pollutant problem, the things that can give us cancer we're exposed to? So here's a graphic I've used for several years. There are 1,000 ticks on this page. Each tick represents a chemical that's been registered by the EPA that is in use for consumer goods of various types. So there's 1,000 chemicals on this page, but if I were to try to represent all of the chemicals that are registered to the EPA and are in environmental, consumer products, I'd need a second one of these slides, or a third slide, or a fourth slide. In fact, I need not just 4 slides, I need 86 slides with 1,000 ticks on each one. So the number of chemicals that's registered by the EPA is 86,000. You see every couple of years, not even a couple of years, every 6 months or so, I have to update these numbers. These are the numbers as of May 2020. But only 2,100 have been tested to see if they're carcinogens. Of the 2,100 or so that were tested, about a third turn out to be carcinogens. So that leaves 83,900 untested chemicals that we know nothing about. We don't know if they're carcinogens or not. So just to do this visually, here are all the chemicals that are registered by the EPA. Here are the chemicals that have been tested to see if they're carcinogenic. So clearly we have to find some other way to speed this process up. It's like the chemicals have some kind of civil right where they're assumed to be innocent until we prove them guilty. And proving them guilty takes 5, 10, 15 years. And what will happen very often is an industry that is making a particular chemical, when scientists finally figure out that a chemical is carcinogenic and it's really, it's meaningful, people are exposed to it, the companies go, oh, my bad, sorry, hey, here's another one, we'll replace it. So that's going on now with some of the chemicals that go into Teflon-like Teflon-like coatings. So it's a big problem. Now, how do we know that these chemicals are reaching us? Maybe we're never exposed to these 86,000 chemicals. Well, here's an article from CNN a couple of months ago that says New Mexico dairy farmer Art Schaap has been milking his 1,800 cows every day for nearly a year, and every day he dumps it all down the drain because it's contaminated, mostly with PFOAs and PFASs, which are chemicals you may or may not know about. You will soon if you haven't. These are polar bears, of course. I was at a meeting in Kyoto over the summer. There were, a couple thousand people there, 1,000 at any one time, and there were probably 200 or 300 scientists who were looking at these chemicals in really strange places. For example, you can find these PFOAs and the PFASs in polar bears. You can find them in penguins, so that pretty much covers the poles. You can find them in animals from the Amazon, so you wonder, how the hell do these things get there? But they do. They persist in the environment. And if these animals are exposed and they're carrying these chemicals in their blood, you can pretty well assume that you are as well. And that's true. Here's a article that showed that there's PFAS contamination of drinking water, which is much more prevalent than anyone has thought of before. You can find in any major city water contaminated with PFASs. The question is, how much is too much? Okay, so these chemicals are everywhere. There's a lot of them. Why are we not deterred? Well, everybody has their own motivation. Here's mine. These are my kids in 1968. That's Ethan with his full head of hair and my daughter, Anna. Here's what they look like now. Ethan's changed his hair a little bit. Anna looks like she's wearing the same colors. And this is Anna's husband, Fred. Why am I doing this? I'm doing this because I don't know if things that these kids were exposed to in 1998 isn't going to cause a cancer this year or next year or the year after that. Most people actually do have cancer to some extent, but their immune system has killed it. I don't know if they're going to get a cancer that finally breaks through. The only one here that's not at risk of generating a cancer or developing a cancer that is caused by environmental chemical is this young lady over here, and that's because she's a mannequin. So how do we identify and predict carcinogens? How many are there? Where are they? That's one of the things we're trying to do. Now, to explain this, I got to go back to Biology 101. So hang on for 1 second. This will be very shallow. You all know about DNA, and there are DNA sequences. The human genome is made up of DNA, and the DNA codes for up to 20,000 genes, roughly speaking, 20,000 genes. And each gene can be expressed, it's called transcription, and turned into this intermediary molecule called RNA, and the RNA codes then for proteins. It's the proteins that do things. Now, every cell in your body has every one of your 20,000 genes. All your genetic code is in every single cell that you have. The difference between cells is, or different kinds of cells, is that different sets of genes are expressed, or they're silent. So for example, cell that makes up your skin may have 1,000 genes spread over the genome somewhere, and those 1,000 genes encode making skin and keratin and all the things that your skin cells have to do. Other cells make red cells, make blood cells. Those sets of genes are different. The ones that are turned on or turned off are different. So which sets of genes are expressed and which are silent dictates everything. And in cancer, there is an unusual set of genes that can be turned on or turned off, and that's what makes them into cancer cells. Now, can we use this information that environmental chemicals or cancers can be identified on the basis of what genes they're expressing and which genes they're not expressing? Then maybe we can tell which chemicals are carcinogens if there's some commonality in that pattern. So, of course, you can't do this experiment in humans, so we do this in human cells. This is a chemical. This is one of the examples of a carcinogen, a very famous carcinogen called dioxin, in part made famous because the KGB poisoned Ukrainian President Yushchenko, and this is what he looked like about a month after that. So lots of genes got turned on and turned off you might not have wanted. But what we do now, and this is an idea that Stefano Monti had, was to take chemicals like dioxin and treat cells with them. Okay, so these cells look a lot like avocados, but they look about the same. They have this nuclei. So what we do is we take human cells and treat them with dioxin, and then wait one day, and then we evaluate all 20,000 genes in those human cells to see which ones have increased in expression, which ones that had been being expressed but now were shut off, or which ones did not change. Now, part of the key here is that it used to cost thousands of dollars to do this. Prices went down to about $400 or $500 for certain kinds of technologies. Technology that we're using in collaboration with the Broad Institute is about $10 a pop. So now it begins to get scalable. So here's a little bit of analog science, analog results. So let's say we treat these cells with dioxin, and then we look at 14 genes. We're not going to look at 20,000 genes because it's hard to picture. You look at 14,000 genes, and you measure whether or not their gene expression is up. So gene 1, after treatment with a known carcinogen like dioxin or something found in smoke, goes up. Red means that the expression level goes up. Gene 2 goes up. Gene 3 goes down. So if you take 200 or so carcinogens, which we did, and treat these cells, you get patterns that begin to look similar, begin to look the same. That is, there's some similarity among all these carcinogens. And then we look at some non-carcinogens. So we take aspirin or something and drop it in here with the cells, and you see a different pattern here. Up, well, that's the same, but nothing changes, up, but this gene didn't go down. So you can see this pattern looks very different. So now we have patterns that tell us that something's a carcinogen and patterns that tell us not a carcinogen. So then you have your unknown chemical, and you say, you treat these cells with your unknown chemical, you look at the gene expression profile, and you say, well, actually this profile looks a lot like that of a carcinogen. It starts off with 2 genes being upregulated and 1 being downregulated and another one being up. So you can see this pattern turns out to be exactly the same as this carcinogen. So that allows us to begin to identify which chemicals, unknown chemicals, are carcinogens for about $10 a pop, whereas the current protocol takes 2 years and about $5 million per chemical, and it involves lots of animals. Now, we've done this with about 300 chemicals, and we find that so far, we're about 83% accurate. We can predict a carcinogen and a non-carcinogen about 83% of the time. This can get up to 90–95% of the time if we had the ability to test more carcinogens. The ability to test more carcinogens depends just on technology. What we would need to be able to do that are these pipetting robots, because you have lots of cells, you have lots of chemicals. You can't do this by hand anymore. You can't scale it up by hand. And what you see on the left is a representation of what the data really look like. I'm obviously not going to go into this in great detail, but just think about this column. Let's say this is one sample that showed this pattern. Now you look at this column right here on the left. That's one chemical that shows this pattern of genes turning on and turning off. So across the top are individual samples, and each row represents one gene that in this case went up, didn't do anything, or went down here, or went down, etc., but went up in this sample. So you begin to see these patterns. Now, you can understand how dense these data are because there's probably only, I don't know, 100 or so rows here, but the actual data go down 20,000 rows because there's 20,000 genes. So you can see the power of this, that even subtle changes in cells resulting from exposure to some chemical, you can begin to find patterns in that. Now, just to give you an idea of the impact of these experiments and show you that part of our charge is to be able to communicate this to the public and to other scientists. This is a paper that was published last year that described this process of identifying carcinogens. So far, it's been downloaded about 3,600 times, about 62 papers that have referenced it. It's in the top 3 to 5% of all papers in this particular area. It was picked up by news outlets and by people around the world. So part of the idea and part of the great work that Find the Cause has done is to get this information out to the public, because ultimately it's the public that's going to make the difference by banging the drum a little bit. Okay, so we might be able to identify carcinogens or predict which ones are carcinogens. Can we now, so if we eliminated those chemicals, that would be primary prevention. Can we now look at the potential for secondary prevention, which is find out who's about to get cancer and then intercept? That's real prevention. No cancer, no precancer lesion. Okay, so what we want to do now essentially is look at what's in this box. What's happening between here and the formation of some lesion, and can we stop things right there? So obviously, it's difficult to do this in a human. You can't start from zero and start exposing people to carcinogens. There is a kind of experiment, though, that I'll come back to in the end, where people will do that voluntarily, and that's called smoking. But we don't want to be doing that to people, so we do this to mice. So here's an experiment where we look to see if we can find the pattern within some organ that's about to get cancer, a pattern that says this organ is about to get cancer before you can see anything else. So what we do is we start with some kind of known carcinogen, like you can find in smoke. Smoke's great, smoke of all kinds, great for carcinogens. There's dozens and dozens of them. I neglected to say earlier on that if I took your blood and sampled it, I'd find dozens of chemicals that are suspected or known carcinogens. So nobody gets off scot-free. So what we're doing here is then we expose mice to these chemicals. Turns out I'm the one that does the exposures because according to my lab, I'm the most expendable person, given my age relative to everybody else. Also, I'm happier if I'm handling the carcinogens and they're not. And it will initiate, in this particular case, lung cancer over a period of about 21 to 31 weeks. Now, you know, we focus on breast cancer, but we find a lot of the answers in some of the other cancers as well. So what we say about breast cancer seems to hold true for lung cancers and oral cancers and other things. Now, I'm going to just introduce, just for a second, this horribly named protein called the aryl hydrocarbon receptor. It is the protein that recognizes within this mouse that there's a carcinogen around. For now, trust me, just think about this as a doorbell. I'll come back and explain that in a second. But once it is engaged by some of these carcinogens, then things start to happen. Now, one day we can take the lung out, and by we, I mean I take the lung out, and then Stefano analyzes it with this computational model. Gail Sonnenschein, I'll show you in a second, is trying to find a really easy way to do this experiment. But in this case, we have to take lungs out and analyze whole sets of genes. This is the pattern that you get one day after you expose them to this chemical. Now, by about 3 weeks or 8 weeks, the pattern changes, and that's because the chemical eventually goes away. But what we can't see in here is that there are things percolating that will eventually result in this pattern showing up again when the full-blown cancers show up. So around 15 weeks, you get a pattern that starts to look a little bit like what you saw one day after exposing to a carcinogen. And then at 21 weeks, right around the time you see cancer, you see something that looks almost exactly like the pattern of gene expression one day after the chemical. So what happens here, we think, is that the chemical starts a process, and in some small subset of cells, that process just stays on, and that's what shows up eventually. That's what predominates later on in cancer. And this is the point where Charlotte Cooper also steps in, because she's an expert in identifying what precancers and cancers look like. So over here, just to repeat what I've said, if you just looked at cancer genes, normal tissue would look like this pattern, and the cancer looks like that pattern, which looks a lot like one day after exposure to a carcinogen. So let's say we start to see this pattern change here at 15 weeks. We know something's about to happen. What do we do about it? Well, then we try to intercept it. And both Gail's lab and my lab have been generating essentially drugs that could stop this pattern through different mechanisms, one of which I'll describe in a few minutes. And that is the point at which we would prevent or intercept cancer as part of secondary prevention. Now, this is a really nice pattern that you see in these mice one day after chemical exposure, and let's say 15 to 21 weeks just before cancers happen. So the question is this pattern that we see in mice after exposing them to smoke anything like a pattern that you can see in a human before the human gets cancer? So what we want to do is ask the question, can you see this pattern in humans as well? Okay, so this was started by an environmental carcinogen, smoke. So where do you look to see if you see that pattern? You look in people who smoke and develop lung cancer. So there's a lot of data out there that's available about the kinds of genes that turn on or shut down in people that have lung cancer, or people that have pre-lung cancer, or people that are smokers and they've stopped smoking, but they still have a very high chance of getting cancer later on. So what we did then was to take, let me just go back one second, to take this pattern. So once again, this is a pattern of genes, about 50 of them in here, that either go down when exposed, when just before cancer happens, or go up just before cancer happens. And we said, what if we take that set of genes? Can we find that pattern in normal human lung tissue or tissue of lungs from people before they get cancer? So on the y-axis here, you can just think of this as the degree to which you can see that same pattern that we found in mice before they got cancer in humans. So in normal cells, that's our baseline. That's how much you see that pattern in normal lung cells, which is in an absolute meaning not much. You don't see that pattern much at all. But if you look at people who have pre-cancer, we only know this because they've been studied for many, many years, and there have been samples pulled out before they actually got cancer. Now you see this increase in the appearance of something that looks like that pattern. And that pattern stays there to pre-cancer and then goes up in spades when they actually get early cancer. So this would be the point at which we hope we'd be able to see this pattern coming up, and that's when we would intercept and try to block for cancer. Now, Gail's idea, in that particular experiment, or in case of breast cancer, we're not going to be sampling somebody's lung. If you're healthy and you go into your doctor, they're not going to be sticking a fine needle in to do a biopsy of your lung or breast tissue. So Gail's idea is to see, can we identify this pattern resulting from changes in the tissue that's about to become cancer? Can we find that in the blood? So it turns out cancers make some of these gene products called RNA. You don't have to know anything about that, but you can detect these RNA in blood. And hopefully we'd be able to find some kind of pattern of which RNAs show up in the blood that correspond to the DNA patterns that I had just shown you. In other words, be able to do a pinprick, take blood and say, there's an RNA pattern in your blood that tells me that you are pre-cancer and we need to go in and intercept. So basically you get the same kind of thing where, here's a normal pattern, here's just before cancer, and here's as it transitions into cancer, and they would be able to say, you are here. So we need to do something right now. Okay, so how are we going to do that? How are we actually going to jump into the black box and do this interception? Now, to explain this, I have to just go backwards one step and explain all of life, which is how cancer happens. Cancer is basically an abnormal version of everything all our cells do over the course of our life. So we're going to talk a little bit about how cancer works. I'm going to talk about cancer stem cells. and then immunosuppression. And you'll see that one of the themes here now is that the immune system plays an enormous role in determining whether or not you're going to get cancer. And then I'll show you some of the tools that we have to then intercept cancer. Okay, so what are cancer stem cells? So let's say this is a cancer. The problem with cancers is they do three things. They grow like crazy. They forget how to die, so they become immortal, as opposed to normal cells who know when they're supposed to die. And they have this identity crisis where they don't know where they belong. They don't know if they're lung cells or breast cells or brain cells, and they go everywhere. And so this is the tumor, let's say, at diagnosis. Within that tumor, there's a very small population of cells called cancer stem cells that exhibit all of these properties. They're virtually immortal, and they just love to go places. So they will metastasize to different parts of your body. Now, depending upon the cancer, this can take 1 year, or it could take 3 years. In the case of some extreme examples, it can take 10 years. So very often you'll hear about somebody coming back from their treatment, and the physician will say, you are cancer-free, but not so much, because this is a very small population. It's really hard to identify. So you don't want to be generating cancer stem cells. That's lesson number one. Lesson number two is we all have an amazing immune system. Unfortunately, cancers know how to turn this immune system off. There's an active process through which they shut the immune system down. What's also bad is that environmental chemicals, which I just told you can make cancers in part by driving cells to become cancer stem cells, which grow and don't die and migrate. Not only do these chemicals induce these guys, but these chemicals also, by themselves, can suppress the immune system. It's been known for a long time these environmental chemicals are immunosuppressive. So this is kind of a double whammy. The chemicals that really are carcinogens because they do bad things to cells, and because in the meanwhile, they shut off the one system that protects you for life, you hope, against cancers. Now, how do the chemicals work? Okay, so now we're going to come back to how cells recognize that there are environmental chemicals around. You know, what do they know from chemicals? Well, think about it this way. Cells determine what's going on around in their environment in many ways similar to the way we do when we're in the house. When you know, the way you know that your Amazon guy has just dropped off your latest package because you're not going out of your house because of COVID. How do you know he's there or she's there? Well, they ring the doorbell. Okay, so that's great. The problem is environmental chemicals keep ringing these doorbells. These doorbells are receptors that are there to do other things, that recognize other things like hormones. And once an environmental chemical gets in there, the bell just keeps ringing. It does not shut up. And that would drive you and me both crazy in the house. And it drives cells crazy. And the doorbell that we have been studying most, that recognizes a whole slew of environmental carcinogens, is this thing called the aryl hydrocarbon receptor, or the AHR. So I'm going to show you how this works, and I'm going to show you how we intercept its activity. So here's our avocado cell again. Here's a nucleus. This AHR thing, this receptor, is out here in this part of the cell. Now, when you're exposed to something like smoke, it activates this receptor, and then the receptor and part of the smoke molecule itself go into the nucleus where the DNA is, and it changes the pattern of genes that get expressed, which we already talked about. And then the way we say this scientifically is bad stuff happens. Well, what bad stuff in particular? As I just said, you get cancer stem cells and you get immunosuppression. So you want to try to avoid this from happening. Now, if you can't avoid somebody from being exposed to these chemicals, maybe you can just block the activity of this receptor. So I'm going to show you two experiments in which we inhibited or reduced this receptor either through gene editing techniques or through a drug that we've generated. So the first experiment is gene editing, where we just take this receptor away and then say, okay, what happens if we have something that looks like a cancer because it's seen this, but now this receptor has gone away? Will the patterns go back to normal, for example? Okay, so this is done by gene editing, it's called a gene knockout, where we go in there and we remove just the gene that codes for that particular doorbell or that receptor, the AH receptor. Now I'll show you what will happen to a mouse, a very aggressive mouse oral cancer cell line once we take this receptor away. So here is a chart. On the y-axis is the size of the tumor. So these are oral cancer. So this happens to be in the tongue. Hope nobody's eating right now. And then we watch the cancer grow over 60 to 70 days. So after injecting it, it takes, 40 or 50 days before we start seeing it, and then it grows like that. But if you remove this receptor, the doorbell, you get absolutely no cancer whatsoever. One gene, no cancer. What's more amazing, and I have to tell you, when I saw this result, I told the doctoral students, the PhD student, I don't believe you. Go back, do this experiment again. Nothing comes out this good. And she's done it 3, 4, 5 times now, exactly the same result. Now, we looked at this and we said, wow, this is so complete. Maybe this is because without this one gene, these mice are now immune to this particular cancer. So we took mice that were out here at 70 days. Actually, they were sitting around for like 100, 120 days before we figured out to do this experiment. And we took those mice and said, oh, let's take the normal cancer, what we call the wild type, the cancer where this has not been removed. A wild, malignant, fast-growing cancer. Let's see what the animal can do with it then. Here's what happens. You still get nothing. So these mice have become completely immune as if they've been vaccinated against this cancer. And just to give you an idea of what it looks like, it's so stark. These are what these cancers look like. This is a cross-section of normal mouse tongue. So the taste buds are out here somewhere, and this is all muscle, because the tongue is mostly muscle. This on the right is what this tongue looks like when there's cancer growing in. So you see all this dark purple stuff that's just growing in all kinds of weird shapes and irregularities. That's a bad cancer. And these are the mice that got this genetically modified cancer first, and then got this fully malignant cancer, and there's absolutely nothing going on here whatsoever. Now, the technology is not quite up to the point where we can do gene editing on a cancer in real time when you have it or when you think you're about to get it. Technology not quite there yet, but the technologies are there to try to identify drugs that do the same kind of thing. So what we've done is to develop a drug that blocks this H receptor. Okay, so, this is, I'm sorry. Sorry, a little bit out of sequence here. Let me just go back 1 second. We're looking now at these mice to see how they're immune. And why are these guys immune? What part of the immune system has been turned on? Okay, so the way we identify how this one receptor is changing gene expression profiles is to look at the gene expression profiles in tumors that have not been manipulated. So these first 4 rows are samples of genes that are being expressed in 4 different mice. And you see the patterns in the 4 mice are very similar. These genes tend to be down, these genes down here tend to be up. But if you knock out, you eliminate this one receptor, you get an opposite pattern where these genes turn on and these guys turn off. Now, by seeing this pattern, we can say, whoa, this gene must be really important because it changes. It goes from going down to going up when you take this one receptor out. Now, it turns out that some of these genes are genes that are extremely famous. They encode for proteins that regulate the immune system. They're called immune checkpoints. They are genes that ultimately result in an immune system turning off. So we said, well, if the mice in which we have put cancers in which this one gene, this receptor has been deleted, maybe those checkpoints don't show up anymore. Maybe this immunosuppression doesn't happen. So this is a result. This just is an indication of the level of one of these immune checkpoints. And in the control tumors, there's X amount of this immune checkpoint. This thing will suppress This protein, when expressed, will suppress the immune system. But if you take away that one doorbell, there's almost none of it being expressed, which means the immune system here is free to go. It's free to attack the cancer. And the reciprocal is also true. If you look at the level of this immunosuppressive checkpoint, here it is in these control mice. You now give them an environmental chemical like dioxin, and it goes up, which means not only do cancers want to suppress the immune system, but also these environmental chemicals want to suppress the immune system. So what we've identified actually is that through this one receptor, that I'll show you in a second, we can block. Through this one receptor, there's at least 6 immune checkpoint proteins that are being regulated by this one protein. It's the kingpin of them all. It's holding up all of these immune checkpoints that are suppressing the immune system in multiple ways. Now, you know about these things because pretty much every night you're bombarded with a commercial about immunotherapy. How does immunotherapy work? Well, these drugs are directed towards, let's say, this checkpoint, this immunosuppressive checkpoint, or this one, or that one. And there have been some amazing results. But people in end-stage cancer, 30% of the people in an end-stage melanoma, their cancer will shrink and most of the time just go away if you shut one of these immune checkpoints off with one of these chemicals. What we're saying is this one receptor, this one protein controls them all. So can we find a drug that we hope would eventually do the same thing all of these do in combination? So here's a scheme of that. Here's our cell again. Here's our receptor with some environmental chemical that has started things going. So keep in mind that by the time you're at pre-cancer or cancer, the environmental chemical is gone, but the receptor is just stuck in the on position like the doorbell. So we've made an AHR inhibitor that just goes in there and shuts this thing off. And here's what happens. So here's the mice, and we've given them, in this case, a very aggressive lung cancer. And then 21 to 26 days later, we image live mice. So the mice are fine, except that the mice that we just do, that are the control mice that we don't give any inhibitor to, any drug to, they develop these lung cancers. So what you're looking at is a mouse that's under anesthesia. This is a little cone that keeps them asleep for a while. They're on their back. His right leg, right front leg, left front leg, and right under there you can see the imaging. So the imaging actually looks right through the skin, and you can see the cancer is growing in their lungs. So all of them, this one's got a bad one going, this one's just getting going, this one's just getting going. But these 4 mice were all mice that got our drug, which we call this thing here. So all that's done is block this receptor. So what we think we can do altogether is identify when something bad is about to happen because of a change in patterns of which genes get expressed, and then intercept using chemicals like this. And Gail has another drug that works a different way that also seems to be effective, at least in the animal settings. So just to finish, I want to remind you that there are ways to reduce your exposure to these chemicals. If you go to the Find the Cause website, you'll see a whole list of chemicals that you should try to avoid. That's only part of it. You want to do that for you and your family, but you also want to be aware that the only kind of change that happens when people start yelling, when people start telling their Congress people that it's time to pay attention to things like the Clean Air Act, which has been gutted, and the Clean Water Act. So it's really up to you to do your part. As Drew said at the beginning, and I mentioned earlier on, only about 7% of the billions of dollars raised for breast cancer research go towards prevention. You will recall my conversation with Dr. Sharpless, where I couldn't get him to actually define prevention. And remember this part: you can make a difference by contributing to Find the Cause. Drew, do you want to finish up with anything, or shall we end right there?
Just want to say a big thank you, Dave, for this incredible presentation. It's so fascinating and it's so motivating. And I want to do 2 things right now. I want to go to my own website, our own website, make a donation. And I also want to help people learn how to understand how they can try to do some avoiding of some of this chemical exposure in their everyday lives. And we do offer workshops in that. So let us know if you have any interest in us coming to you either virtually or in person and doing one of those workshops. But most of all, this research is incredible, and it can only go further as if we really do get this movement going and really fund prevention to the level it needs to be funded so that we can make this sort of progress. So again, thank you all for attending this interesting presentation. And David, thank you for your incredible work, to you and the entire consortium.
Pleasure. Thank you.